meiosis. So same deal with cells that go through meiosis. They go through the cell cycle. We are not making you memorize every single step of the cell cycle. You are welcome, but just know that cells are always going through these little checkpoints. They're duplicating their DNA and going through these stages so they can split and reproduce. Now, the difference with meiosis and mitosis is that the purpose of meiosis is to create genetically different cells. This is used in reproduction. So this is how women get their egg cells and how guys get their sperm cells. So how do they end up being genetically different? It is due to this process called crossing over. So when the cell is duplicating its DNA and getting its ducks in a row, the homologous chromosomes will align with each other and they will trade little pieces of themselves. So when they eventually go to split into different cells, the cells will not be genetically identical. They get these kind of mixed up chromosomes, so the offspring will therefore not be genetically identical, which can be useful in terms of evolution. This allows for more genetically diverse offspring, which can be good if you're trying to adapt to a new environment. All right, so meiosis is divided up into two stages. The first one is called meiosis 1. So in this one, the homologous chromosomes are going to line up and they are going to separate. And this happens after crossing over. So you can see here in this little picture, in prophase 1, they're just floating around. Metaphase, they line up, and instead, so in mitosis, sister chromatids separate first. In meiosis, homologous pairs separate first, and you can see that there. All right. Now, in meiosis 2, you have two cells now with the separated homologous pairs. After that has happened in meiosis 2, then the sister chromatids separate. So you end up with 4 genetically different cells instead of just 2. And each resulting cell has half the amount of DNA as the original did. All right, so let's draw it. Prepare for your hand to hurt a lot worse this time. All right. So, drawing a sex cell before it is ready to split, we are still going to have our 46 chromosomes, still going to have 23 homologous pairs, and still have our 46 sister chromosomes, our sister chromatids. My only difference here is I'm going to make the chromosomes from dad one color and the chromosomes from mom one color. So I'll make the chromosomes from mom blue. So I need 23 from mom, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. All right, and I'll make the ones from dad green. Actually, no, I'm not. I'll pick a darker green, actually. All right, so 23 from dad. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18, 19, 20, 21, 22, 23. All right, so at the end of the day, each one of those squiggles is one chromosome / cis chromatid, and then the pairs are still pairs. All right, so let's get it ready to split. All right. So we're going to do the same thing that we did before, except we're going to make our homologous chromosomes line up. So what did I do for mom again? Mom's just blue. All right, so I'm going to draw 23 of duplicated mom's chromosomes up on top. So I'll draw little X's for each one. So 1, 2, 3, 4, 5, 6, 7, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. And I'm going to draw 23 of dad's duplicated chromosomes on bottom. 1, 3, 4. All right. Now, let's draw the crossing over process. All right, so in crossing over, all these chromosomes are going to exchange little pieces of themselves. So this top of the green one might go to the, oh crap, I just erased it. Whoops, go back. Okay. my bad. All right, so the top of this green one might go to the top of this blue one, and the top of that blue one might go to the green one. The bottom part of that green one might go to the blue one, and vice versa. Okay, so all of these are going to trade little pieces of themselves now. All right, so just go through, add some green to your blue. Doesn't have to be super perfect, just get the idea. And then go add some blue to your green. All right, so now we have our genetically recombined chromosomes all together. So we still have 46 chromosomes. We still have 23 pairs, but the chromatids have duplicated, so now we have 92 sister chromatids. But remember, the sister chromatids do not separate yet. The homologous chromosomes separate. So we would draw that like this. So up here, this entire first row, the blue with the green drawn on it, all of that is going to go into this one here. So let's draw that. Let's draw our 23. 1, 2, 3, 4, 5, 6, 8, 9, 10, 11, 12, 13, 16, 17, 18, 19, 20. All right, and then they have the green splashes in them, so we'll add some green real quick. All right, and then all of the chromosomes from the bottom half here are going to go into this one. So we'll draw some green X's. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 20, 21, 22, 23. Then we'll add in the blue. Yay. All right, so now we have some separated cells. Now of these, they each still have the 46 chromosomes. Now they no longer have any homologous pairs, and they still have 46 sister chromatids. Actually, my bad, that is supposed to be 23 chromosomes, because each one of those individual still counts as a chromosome. It's just the sister chromatids that are duplicated. All right. So now going on to meiosis 2, this is where the sister chromatids do split. So each one of these X's, half of the X is going to go to one cell and the other half is going to go to the other one, similar to mitosis. All right. So we'll split up this one here first. So this cell, again, half of that's going to go there, half of that's going to go there. We'll put them into these two. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. These still have a little bit of green in them. I'm not going to do all of them, but you get the idea. And then in this cell, half of it's going to go there, half of it's going to go there into these two. So 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 19, 20, 23. And then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. All right, and then we'll add some blue. Add some blue. You get the idea. All right, so now our end result is the 4 genetically different cells. So, and now in each one of these, we are left with 23 individual chromosomes, still 0 homologous pairs, and now 23 sister chromatids. Now, if let's say that one of these is an egg cell and that one of these is a sperm cell from another person, those two cells would combine, and then you would get back to the normal 46 chromosomes that make up everybody else. So I hope that